when that's still.
Yeah. Might want to.
Yeah, you're right.
Yeah,
That's what I was
just
going to do.
straight as, nice.
There we go. He's in there.
Great. Thanks so much.
All right. Tracting drawer.
This is 04 Bio. Thanks, pilot.
Yeah, just send my came in. The manip cam's really good for this one.
Copy that.
Yeah,
Nope.
just a little offset.
Right there. Oh, no.
And we have our
That looks
friendly
good.
wreckfish
You just gotta get in a little
checking
bit,
it out.
and then you can release, and then just
Kind of push in
the
there?
rest. Yeah.
Like that? And then I can release?
Yeah.
Okay.
All right. Then you can just close and just give it another tap in.
Okay.
And she's in.
Okay.
Very
job.
nice
You did
scooping,
great.
pilot.
Thank you.
Do you want me to come up with mini Zeus?
Yeah, sure.
All right. Coming up. Stopped. Nicely done.
Okay. There we go. We're secure.
Ramping down. Steal my camera back from you.
And I'm going to take my cameras back too.
Yes.
And if you want to bring them outboard again and store my
Oh,
wings
the wing, yeah.
for flying. This guy's just checking us out today.
Yeah, he thinks you're just an old friend.
Yeah. Get out of here, fish.
All right, moving outboard.
Okay. I think we're ready to take off and continue.
Yeah.
Great.
I have you about 75 in my camera.
Perfect.
So we've just finished our first geological collection of the day, which was some of the coral rubble, which will be used for dating, and
those-
some sediment,
those
which will be
fish.
used to examine what is making up the sediment or sandy matrix that we see all the coral rubble on top of. And we also collected a hydroid, what we think is a hydroid, but we're not sure, and an urchin. And those are very good representations of what we've seen today. And we have an hour and 45 minutes or so left on the bottom. We are trying to make it up to a depth of about 375, and we're at 385 meters now, so we're making good progress. Pilot, can we look at the yellow, I think they're corals in the upper left?
Absolutely.
Good
Let's go and get
snapshot.
lasers on. for
Yes, lasers are
stuff.
coming on. There you go.
You're talking about right above the lasers, correct?
Yep, that's the one.
Copy that. Let's do a snap zoom video while I make my way over there.
Huh. That's different.
So are we going to want a closer look at this? Because I'll go set up for
Yeah,
it.
yeah, I would say so.
Alright,
I'm not
come on out,
recognizing
video.
it. Let's see if anybody else recognizes this. They're quite a mustard
Do you
color.
think where you sit really close to it?
Yeah, I'll do what I can. Gonna have to land on.
Yeah.
Go ahead and video.
Did you guys just lose a light? Because I didn't change the iris.
That wasn't you? Stand by.
It wasn't me unless it was a delayed reaction.
My manips never changed lighting, so
Okay.
I don't think we lost one.
Maybe I lost lens communication momentarily.
Okay.
And this is
Yeah, I
an
don't see any
octocoral
faults
for
or
sure, and
So I don't think it was
nobody
the ROV.
seems to recognize it.
Do you want to try the fog lights for this guy?
Yes,
In-
please.
Okay.
All right.
Should be a good placement for it.
Fog lights coming on.
Yeah, we're just a little shy of it.
Just a little too far out. You want the tips of it so we can maybe...
Yes, please.
Okay. Like up here?
Yep.
This looks better. Oops. I took off a little bit there.
And go back down towards the base.
Sure. We can definitely set up for that.
So,
That's good.
since none of the scientists recognize this coral,
I think for
we
setup
are going
for the
to try
collection,
to
I'm gonna
set
wanna
up
get
for a
little
collection.
further in.
Yeah. I
It's
agree with
definitely
that.
growing on dead skeleton matrix from a stony coral. That's a great shot, and that little bitty squat lobster. He's still giving it his all.
I can't believe he's holding on right now.
Full screen.
Okay. And let's kill fog lights as soon
Yeah.
as video's clear.
Go ahead and kill the lights.
Lights
And
coming on.
let's fill the screen.
And get rid of lasers.
Lasers off.
Lasers off.
Thank you.
Watch lead nav.
Video's clear.
Copy that.
Go
Come
ahead,
on
Nev.
out. Video. I'm going to
Just
set up for a
be
little.
advised, we have
lecture here.
120 minutes on the bottom, and if we headed straight for it, it would take us about 30 minutes to get to this peak.
If I
the
set
last
up
waypoint.
for,
Great, thanks very much. It sounds perfect.
are we going to be able to get our
I'm trying to think.
skids out?
So while we're
It's-
setting up for a collection of this
Maybe.
unknown octocoral,
Well,
we have
worst case
about
scenario,
an
I can
hour and
move.
20 minutes
Yeah.
left in the dive, and approximately a
close.
half hour to our last
This
waypoint.
fish is
So
back again.
we can take our
There's two
time,
of them. I think they're just circling you.
and here he
Probably
comes,
waiting for a moment of weakness.
photo bomber.
He's making loops around you.
Okay. I don't know
He's
if we'll be able to come
stalking
out or
us.
I
not
know.
with that right
I'm
skid,
starting to
but
get a little creeped
see what
out by him.
It's possibly because
You
after
start
the fact
dreaming
rotate
about it.
a little
Glad I'm not a
starboard.
crab down
I think-
there.
Yeah.
Yeah,
after the
Or
fact,
a
rotate a little
squid.
starboard and then,
they might.
okay.
We'll see.
Okay. I don't think that's too far out, do you?
I think it'll be fine.
Yeah, I think so.
from serious view, he just turned at the blue lights and looked like he was coming back around.
So this is
again.
going to have to be a starboard.
Yeah.
I'm gonna go for the starboard in there.
Yeah, it's fine.
Okay.
We've heard that he is circling us.
You're ramped up. Ready to go.
Excellent.
Starboard.
Hydraulics on. Grip force. I'm gonna change down to a 4.
Are we getting a snip or the whole thing? Because one of the comments is the cross-section of the stem would tell us some information.
Yeah,
Okay. So what are you guys thinking, watch leads?
I would say we should probably go ahead and get as much of it as we can, since this
Yeah.
might be new and that might help us identify it, and
Yeah.
there's 2 more of them here.
And the current, if we just get a snip, it might not stay in the basket anyway. So.
Yeah, correct.
Okay.
So I'd say grab towards the base if possible.
Okay. Could we do a quick zoom in video on the main?
Do you want to center your mini Zeus?
Yeah. Well. Yeah. Go ahead and do that while I'm looking at this.
Copy.
Okay. I think if I can get that chunk there.
Pilot, it looks like it's growing on a stalk of old scleractinian coral, so I don't know if at least part of it is, but then there's more coming over from the right, it looks like. So
Okay.
it's a complex situation. It might be 2 of them, actually, but
Okay. Well, I'll grab as much as I can of this, and
Yeah.
we'll go from there.
that sounds great.
Okay.
Thanks.
Come on out video just to get my manip in there. Perfect.
They are pretty small when you look at it compared to the manip.
And as Scott France points out, the stem can also give us some aging information, the base of this. And some of these corals, Cheryl, the ages of these, some of them have been dated for being hundreds of years old. Maybe
Or
not
even
this.
thousands. Yeah, the
Even
black
thousands,
corals.
The black corals.
There's one off of that was collected off of Hawaii that is over 4,000 years old.
Well, I got a lot of it.
You
That's
did.
great. That's great.
You certainly did.
All right, let's go ahead.
That's
Ready
great.
to extend?
And
So
you
we'll
can-
be
you
able
can come
to
out
use
wide
this
on my-
for many different examinations, taxonomic
ready
ID,
for-
and possibly
coming out.
aging.
Extending.
Chemical composition, for sure.
Hey, look, there's the other guy. He's going to fall in.
That'll-
Boom.
We got a
Look at
bonus.
that.
Just like you planned, right?
Going
That's
for a
funny.
record number of samples for one pilot seat.
So this is probably in
Nicely
then.
done.
Okay.
Yeah.
Well, it was in the inner, but it's a lot easier for me to get in the inner. I can certainly try for the outer, but I mean, I'd rather go for the inner if you guys are okay. But we do have that other sample that fell in, so it's kind of whatever you guys would like. Watch leads.
Well, I guess we'd rather have both samples. So if they have to be in the same one, then
I don't think we should combine 2 samples from very different locations either.
yeah,
In terms of
it's-
associates.
Okay. I'll go for the outside, and if I'm having problems with it,
we can reconsider.
we'll reconsider. Okay.
Yeah, that sounds good.
Might be big enough. It might
Yeah.
fall in because it's weighted.
That's what I was thinking too.
So might
So-
not have to physically get it in there.
Okay.
Do you want to rotate down?
Yeah.
Copy.
You go ahead and keep me. This is going to be different. I'm in there.
Yeah, you're in there.
And it's in there.
Very good.
Close
We are
draws.
bio boxes full.
Oh, yeah. I
Nice.
And this is
thought I was. Thank you.
Bio 5.
Just give me a second. I got to get this guy.
Yep, no, they're sunk. In a good spot. All right, retracting.
Once
door.
again,
Okay, there we
great
go.
job, pilot.
Good job.
Okay, if you want to follow me up. Are we zoomed out on there?
For Mises, I
Yeah.
think we're full wide on it.
Copy that. Thanks.
We're having a hard time seeing any rocks today.
Fortunately, I wasn't expecting any. So I'm not disappointed.
We're not done yet either. So.
Yeah, that's right. Truth be told, I'm
Okay.
more happy with sediment than rocks.
Secure.
I'm that
Nice.
kind of geologist.
Backpack come on.
Yeah, that really helped that it was heavier, so it sank in there.
That's great.
You're
You did
right.
a great job there. Tucked you right in.
And our bio boxes are full. We got some really nice samples.
Oops.
Steal
And
my
this one is going to
HD
be
bag.
extremely
I'm
interesting
gonna
to
set up
take a
mini,
closer look
and then I
at.
think we're shifting out, so it's a good place to just...
I didn't see any
Yeah,
obvious
shift.
associates. Did you when we were
No.
zooming in? That would be
I
interesting
think
to
I
see.
saw a possible anemone attached to that.
Oh, to the
coral.
base, maybe?
Yeah.
Okay. Hello,
Yeah.
do we have someone calling in?
And one small chunk of that one fell into the inner box.
Okay.
Hi, it's Nolan.
Hi, sorry, we couldn't hear you.
Hi, it's Nolan.
Oh, hi, Nolan, how are you?
Pretty good. How are you?
Great.
I figured I would just make a little comment about the sponges now after we've gotten all our collections for the day. So.
Sure.
Okay, so first of all, I got a look at, I did some more digging with some of the sponges. So I definitely think we're seeing Geodia pachydermata, like Shirley Pomponi was saying. And then it looks like there are more Geodia that we've been seeing, and I just found that the genus of Geodia has about 200 species in it.
Oh, wow.
So
I'm so sorry.
so the pale, round, smooth-looking sponges are also Geodia.
Really?
And
Okay.
they look like, they look something like Geodia notistrella or Henchley. I think that's how to pronounce it.
Huh.
And so the sponges that you asked me to look up initially, the sort of like fuzzy base-like ones,
Rounded
sort of
face.
like,
Oh, no, the ones, were more vertical. Mm-hmm. Maybe like the one in the
sort of
field of view now?
sorry, go ahead.
Video?
Again?
Like the one in
We're
the
doing-
field of view now?
Doing well,
Yeah.
pilot. How are
Okay.
you doing?
Good.
The upper right-ish.
Yes.
Yeah, so those look something like
Or just don't
a
clip it out as a
solid
sample.
glass sponge, and it looks something similar to Acanthacus or Acan.
Yeah, I should ask them because that was one of the original samples.
something like that.
Mm-hmm. There's a whole
And
cluster of sponges
I'm not
coming into
really
view
sure
right now.
because, so the images we have of it in the animal guide are from the Pacific, and I have not been able to find any information on it in the Pacific or in the Atlantic.
Go ahead,
And
Wesley.
of course, neither Shirley Pomponi or Joanna Xavier are on the line. Ooh, yeah, I see more of them. Okay, I think I have like a 10-second delay. But since we've
Alright, but
been
you can come
seeing
in on
them
the spot.
in multiple
Watch lead, this
dives,
is Nav.
maybe sometime in the future we could think about collecting one of them just so we can figure out exactly what it is, maybe?
Watch lead, this is Nav. So I'm just looking at
Zoom out a little bit, Roland. There's
a high pack here.
two of them right next to each other. We
The
can grab
vehicles are only about
these guys.
80 meters from the top of the feature at
Yes,
0.5.
this sponge that you're
but we
now
have
zooming in
about
on, yeah.
come
I
in
have a
roll
little bit of
on the
delay.
double.
over an hour and a half left on the bottom. So I'm just wondering
This
what
could be
you were hoping to do
in
to
the
use the
family,
time.
Hexactinellid family Rostelidae.
Well, thank you, Nolan. Just so happened that we saw this little cluster, so you get to talk about the sponges while you're actually looking at them.
Okay.
Yeah,
All right,
no, that's
so
pretty
we'll
cool. And
make sure we
if you'd like,
explore
I can talk a little
thoroughly
bit more
and
about just the
slowly
general
so that we don't reach
taxonomy
the top too
of
quickly.
sponges for the audience.
Okay. Maybe we'll continue to move on while you do that, but please, yeah.
Sure, that's fine with me. I've gotten
Video's clear.
plenty of
Thanks, video.
screenshots that I can send
So
off
let's
to the real sponge experts. I'm just a sponge novice.
take our time exploring
So-
the tether length, and we'll move the ship.
So to begin,
Every once in a while.
the
Understood.
audience mayor and youth scientists on board have been hearing a lot of us mention hexactinellid sponges and demo sponges. So there are three classes of sponges in the group Porifera. So Porifera meaning holes, lots of holes, and that's why there's sponges. So there are three classes. We have the calcarea, the demospongia, and the hexactinellida. So the calcarea, their spicules, so these little tiny pointy structural elements, sometimes not pointy, are made of calcium carbon. And they tend to be really small, and their spicules, if they have points, of between 2 and 4 points, and since they're so small, they're harder to find, and they're usually associated with shallow water.
Large
The
spotlights
second
on the
class,
top is in a good
demospongia, which
position of the
encompasses
image if you
between
want.
80 and 90% of all sponges, just because they have this really successful body plan that allows them to get a lot bigger and absorb nutrients by filtering a lot more water all at once. And their spicules
Could
can
frame
be
these
made
guys up
of
if you want
both
it. And I can spin
silica
around them.
as well as a protein named spongin. And they have skeletal fibers that help structure this more complicated body plan that are made of the polymer chitin, the same molecule or the same polymer that's found in arthropod shells, as well as the structure of fungi. And
gonna
they
range your sonar
can
in.
both lack
Pilot. Okay.
skeletal fibers
30
and
meters.
spicules, so
Sounds
it can
good.
make them a little more fun to work with. But, if they have these or they don't have spicules or skeletal fibers, that they're probably a demo sponge. And then the last, and I think one of my more favorites, the hexactinellida, they're made almost entirely of silica, silicon dioxide. And the reason why they're called hexactinellid is
It's
because
like right when I
their
bumped the ground, I
spicules
think.
almost always have 6 spikes or 6 points on them. And that
All right,
allows
video,
them to
maybe
sort
going
of
on the
hook
scroll while
together
you're looking at
and,
us.
and make the beautiful grid-like patterns and shapes that we're seeing on a lot of the sponges. So that's just a little introduction on
Very
that.
cool. I
Understood.
was trying to write that down so I would remember, and then I realized I'm never going to remember this. So you'll just, that's great. And I hope you'll still continue, I know you will, continue to contribute to the chat room. And maybe when you identify something, let us know which of those groups it belongs to now that we've heard, especially the different compositional groups of the
Sure.
chitin,
I'll definitely put it in
calcareous,
the chat room so
silica.
you guys can have a less fast
Okay.
or a way to get back to what I said.
Yeah,
Okay.
video.
Great.
And I'll definitely
Thank you.
call that more often. I'm home in California on a little bit of a vacation.
Oh, okay.
So, I have more opportunity to call back in. So.
All right. Well, thanks, Nolan.
Thanks so much.
And I'll see you in Charleston at some point.
See you soon.
All right.
And I have
Appreciate you calling in.
I have seen so many close-ups of the Eumunida pictus squat lobster, and that might have been the most gorgeous one ever.
That was
As
spectacular.
we
zoomed in on
going
this
in really
community, if you were watching, so many of the anemones suddenly pulled in their tentacles
right
all at once. It was beautiful.
Right. If you want to do that, I recommend the right side where all the anemones were. It's very colorful.
Good. Watch one. Yeah. Okay, we can do that.
This is a beautiful community. So this is the same soft coral that we just collected.
I'll hold off until you get closer.
Correct?
Okay. Cecil definitely.
Yes, I think this is the same type that we just collected.
Which has stumped our scientific party online, the chat room. So.
And then here we have an associate.
Wow. It's a very large ophiuroid, or brittle star.
Yeah, you could probably come down a couple meters, Lars.
Coming
down. He's pushed out ahead of you. You're looking down at 40 degrees. I think like a 15-meter delta is acceptable here. Yep, you'll still just be at 12-meter altitude, which is good.
Yeah, and so we see these astrochemiid
Pilot's clear.
ophiuroids on a lot of times with different species of corals, and since we don't even know what species of coral this is probably the first time this association has been videoed.
I wonder if the size of the ophiuroid correlates to the age of the coral, because if that's the case, then this must be a pretty old piece of coral that's relatively slow growing.
Yeah, that might be true, but we usually only see this type of ophiuroid on a coral, so I don't think that they move around too much. So this one might have found this coral a long time ago, and they've lived together ever since.
We'll try to get close.
Yeah, I
to the
remember
still camera.
Scott France telling me about this sort of relationship, I think back in the Pacific, and he said that they start, or the corals tend to find their ophiuroids, or maybe the other way around, really quite early on, because we've seen some very small corals with their associated
you can turn on so you can see
ophiuroids, and then other corals with their associated other
the lights.
Yeah, you can bring the lights on.
It probably could be that they've been together for a long while.
Yeah.
Can you put it up on this monitor 12, Lars? I'm not sure where it is here. Oh, here. No,
There's actually a Bathanectes swimming crab underneath this dead coral skeleton. There's so much to look at here. It's pretty amazing.
It's the bottom row, kind of in the middle. Yep.
At least a half a dozen the Eumunida pictus squat lobsters.
Did you see the stills that we got of the rake or the wreck fish?
I did, yeah.
Yeah, we got quite a few.
Looked good.
How about a slow pirouette at the same distance around as the still camera's doing its thing?
Yeah.
Just use HD2 to know where your skids are.
Gotcha.
You could actually... Yeah, there we go.
Just taking a little time to get some beauty shots here.
I think we should take some time for that beauty shots. This would be the place. Look at all that color and life. It's amazing.
A bouquet. It's really amazing how clustered and
try to get
dense
even closer.
it is.
Yeah.
Understood.
Especially those anemones.
The more colorful side is the other round to the right. Did the coral actually
That's
facing
good,
us?
Levi.
Yeah.
That looks like it's in focus there. can see it's like just at the edge of the toes.
It's so close. Alright, spin around the other way, rolling.
You're still on the back side of the corals.
That was as far right as I could pirouette, I think.
Yeah, getting some good ones. This looks great.
Alright, now I'm going to go for brittle star here.
Looks more like a squatting house than an oasis to me.
So this is very different than anything we've seen so far on this dive in the concentration of life here on this one set of coral rubble. So we have a species of octocoral that we're not quite sure what it is. We just made one collection. One of those has an ophiuroid associate, which is a common association that we see with other octocorals, but since we don't know what this one is, that's a very unusual observation to make.
And it's also a brittle star that we don't see throughout the rest of the area.
We've seen
common.
Millions of brittle stars today, and none like that.
The very small ones instead.
Do you want to get lasers on that for size?
And then we have all sorts of anemones just covering this dead coral matrix
yeah,
here.
you can turn them on. Just check with pilot.
And then we have the squat lobsters, Eumutidopicta, and even a swimming crab on the bottom of this pile.
It's kind of nice, you know, you can
it's just
ask pilot if he wants them on or off, but just use your best judgment, you know. I think most of the time we want to have them on, except if there's like a really nice shot that the highlight editors might be able to use.
Yep.
Just go ahead and turn them off for a few seconds so they get a good shot.
It's just amazing.
We haven't seen our friends, the wreckfish, of late. Maybe they gave up on us or decided we weren't going to be too threatening, but we are still seeing a lot of squid in the water column above the ROV.
Wow.
Sirius view shows that. Maybe they're feeding now and not worrying about us.
Mm-hmm. And we should be getting to the crest of this mound. We're still going up. We're at 384 meters.
You want me to give you
the stand of coral is getting higher.
Like 20 meters?
Yeah, I think so.
Just ahead at 145?
Yeah.
And from previous dives, I actually did a dive on this very site, but coming up from the south side of this mound, so the other end. And once we got to the top, it was just covered with Lophelia, so we'll probably see that. On the south-facing side, there was more live Lophelia further down the mound, and that's something that we're seeing in general. Different scientists have contributed.
20 meters, that's 20 meters, bearing 145.
Most likely because of the Gulf Stream's influence, and that side will get the full brunt of the Gulf Stream current, where we're kind of tucked in the lee of the current a bit here. So they have a faster delivery of food, and they also have the current will take sediment away. The corals can only handle a little tiny bit of sedimentation, so that's something that may limit where they are able to grow. That looks like a nice little cave.
It does.
Want to look inside?
Copper bridge,
Fog
thanks.
lights, yeah. Just hit those reach in there.
Fog lights?
Yeah.
Okay, coming on with the fog lights.
Fog lights won't reach in, they're pointing down.
Oh, okay.
Should we try
Maybe one of those larger
But I can
crabs
zoom
that we
in
saw
from here and
earlier.
light it when I'm tight.
All right, video. Let's see what you got.
We are certainly not hurting for squat lobsters here.
Tilt up off the light.
Seeing if anybody's home. Don't think that anemone is what made this.
I don't think so. My thoughts exactly.
I don't think anything's in here rolling.
Some other tubes of tube worms, and you can see how the one in the distance has sponge spicules glued onto its tube. They take whatever is available from the sediment to build their tubes.
What would you do if a graboid came out of there?
A graboid?
I'm not sure what that is.
Sponge spicule is a good defense mechanism, I think.
Let's reach back to some
Anyone
sci-fi.
nipping away at them.
Oh, I know the reference.
Fish tail in the upper
Yeah.
section. Zoom from here.
Yeah, snap. Like.
Well, there's somebody. Ooh. That was long.
Wonder if that's a conger eel.
Yeah, that was long.
And that would be another likely suspect in. Yep,
There
there
he
it
is.
is on the other side, conger eel.
Yep. You see the tail at one end and the head at the other.
Think he's wise to you.
Yeah. I think he's hiding from me. We'll let it go.
First one we've seen today, but we have seen them in other similar environments. We are really chalking up the fish species today.
That's Conger oceanicus, I think. But I'm not a fish guy or girl, so.
Oh, now the head's out. He turned around in that small space.
It might not be as small as we think.
Yeah, it's probably.
Snap on his face.
Yep.
And wow, are we
Quite a bit.
continuing to see anemones?
Wow.
I think that's the closest I've ever seen a conger eel. That's amazing.
All right, video. Let it go.
Those pink corals at the bottom.
Nice little neighborhood.
All right.
I'm looking down to at 65.
Okay.
Copy bridge, thanks. Move is complete, pilots.
After that.
10-4.
The density of these
those
anemones
redfish
is
are still up
astounding.
behind you.
They are clinging to every
Hanging
square centimeter of that dead coral branch or that framework. And the density is certainly increased facing into the current. You can really see that in this view. We're looking now along the southward facing slope of this mound. It's quite steep. We've been working our way slowly up the backbone of a ridge that leads up to the crest of the mound. Our approach has been from the northwest, moving southeast, and then around to the front of the southward facing slope. And there's another wreckfish. We've seen a lot of these today, if you're just tuning in. They're at least 1 meter in length. And they seem to like this habitat quite well. And there's some other fish down in the lower slope there that might be out of our reach. I don't think it's anything that we haven't seen.
The other fish getting awful close to him. He didn't seem to be a fan.
Although it does look different.
That's not a black belly rosefish?
Well, I can't tell if it's that pinkish color or not.
Yeah.
It may be.
Oh,
Oh, no, I'm not
There's
talking about the
two.
one in the laser beams. I'm talking one now at 3
Yeah.
o'clock, but I think that's also pink, yeah.
Yeah, they think that's the black belly rosefish.
Yep. I wasn't even looking at the one right in the middle of the screen.
There's so much to look
Yeah.
at. And Ken Sulak confirmed what I said earlier about the black belly rosefish, that it's kind of an unfortunate name because there's nothing black on it on the outside. And it gets that name because if you were to open up the fish and open up its gut, the inner lining of the gut is black. So it has a black belly on the inside, but that's not a very good name for being able to identify it from the outside.
Well, good thing, once you know it, it's easy to recognize.
Yeah.
You don't have to cut it open to identify it. And it is quite common.
Especially with Lophelia.
I'd be there at 50 degrees.
Copy that.
Now we're seeing higher densities of sponges than we've been seeing on the lower slopes.
You're going to take lasers off.
Okay.
Same
Seems like
species,
we're
I
seeing
believe,
everything
but more
in
of
this
them.
area.
Sounds good.
This one
And
baby
Joanna
shark.
says that they might be Vizella portiolesi.
Rossellidae, the group.
They seem to like it right here.
Lots of Venus flytrap anemones and, of course, the other pink anemones, pinkish coral-colored anemones that are strikingly abundant. Okeanos urchin passing our screen.
Good, watch one. Yeah. Yeah, understood.
We're going to climb a little. We've got to get up to the
well,
crest.
actually, we actually just put in a ship move, so we have been moving. And we're just about at the top. We're at 380 meters depth, which is our last contour line before what we estimate to be the peak. So, yeah, we'll keep moving ahead, but we got plenty of time.
Another Bathinectes swimming crab.
Go ahead and put in 20 meters.
Yeah, quick zoom on that.
I'd be there at 40
All right,
meters.
Snap on.
Degrees.
Go bridge.
Two of them. I wonder if that was another mating situation.
Copy. Now's a good time to do
or
that.
preparation for that.
Copy. Let me know when you're done. We'll put in a move. Thanks.
Foreplay.
Or post-play. We both sort of hesitate before we say things like that. But they look happy.
Yes, this is for sure.
You can come in further if you'd like.
I think he's winking at us, actually.
I think so.
Seems to have a single claw down and, or I'm sorry, leg down and under to hold that one. They seem to be quite peaceful with each other.
Some very happy crabs.
All right.
With some very
Path's clear.
dangerous claws. Dangerous looking. Really steep here for the last 5 or so meters to the top. It's like an anemone carpet.
Right, straight ahead.
Ooh, and here, this might be another Rachinia spider crab coming up.
Copy that, Bridge.
Couldn't get it from
We'd like to put in
down
another move now.
Lower right. We saw this.
Yeah, we'd
guy
like to
from
do
the
another 20
backside
meters. It's
before.
20 meters, bearing 145, speed decimal 2 knots.
Is this what you want, Roland?
Yes, sir.
Come in.
Good copy.
Partial.
I'd be there at 25 degrees, pilot.
Copy. Coming down, Roland.
Get a tilt.
This guy has such long legs, he can walk right over the top of that prickly sponge without even touching it. Oh, it looks like he's missing one of his front appendages as well. And lots of barnacles on his back. Look at all those.
Wonder if those barnacles are an indication of how long he's lived. Do we have any kind of date timing on these guys?
The anemones close up as the squat lobster backs up.
Crabs are having a face-off here. Can wager some bets.
I'd be there at 5-0.
Okay.
I think the squat lobster is too close for the spider crab to get him with his claws.
Yeah, that's true.
All right, but if you want to come in on the face, maybe. I don't know if you've already seen this or not.
Little tilt up to the horns.
Some barnacles. See these on all the crabs when we take a close look. Boy, he's got a lot of them.
Yeah, it sure does.
Okay, video's clear.
Pilot's clear.
So we've talked about a lot of the corals and sponges being pretty old down here, but because of the cold water and the slower metabolism, as we can see by this crab, a lot of the other organisms can get pretty old down here too. I mean, this guy looks like he hasn't molted in quite a while.
Yeah, that's true.
Yeah, that's a good observation.
I have you there at 7-0 pilot.
I'm not sure about the life history of these Rochinia crabs and how old
Survey
they're
one
estimated
staff.
to get.
I was actually trying to get a hold of
Me neither.
Survey
I
1
just
next
thought
to
it
you,
was something
to your
kind of
left.
cool. Sort of interesting that not just the sponges and corals, but the crabs and the fishes and all sorts of other organisms are just, it just takes a while for them to
Copy bridge, thanks.
grow up.
Like some of my students.
Hi.
I'm not
hear that
including
were
you,
interested
Nolan.
in the operating frequencies of the USPO
I don't want to ever
system.
It's overrated.
So I was just looking at the manual here, and for
I don't
our
know what it feels
transceiver,
like.
it looks like
Yeah,
it gives a range of
this is my
7.5
playground, watching
kilohertz to 12.5
Well.
kilohertz. I don't know if you need
It's a great one to have as a playground.
more specific information than that. Okay.
Indeed.
Yep.
I have you there at
You guys have
8-0
actually made
pilot.
students of us engineers as well. You guys have taught us so much about all the biology and the geology over the years.
Well, Roland, even us scientists have learned quite a bit from you engineers and pilots and everything, so thank you as well.
Yeah, it takes the whole team to be able to do this,
Yeah,
so
now that
we're
you're
all
looking down
very
at him,
important
I would come up
parts.
to like 18.
And it's so much
meter,
fun to have
delta.
all of the expertise both here on the ship and then on shore as well to contribute to making these dives just so much more informative than they would be with only a portion of the participation.
And we're looking at a conger eel.
And that move should be complete now.
That's one of the great things about the telepresence
He's
system
called down
that we
upward.
use. It
Yeah,
brings so many professionals together on what otherwise would be considered a small ship to house the 60 to 90 people sometimes engaged.
That's true. I think that Casey gave us a statistic that we've had over 100 participants in the chat room so far this cruise. So I think she said 102. So that's, I mean, yeah, you couldn't ever fit this many people on one ship. And then we have people that have called in from all parts of the world. And that's just such a great situation.
Even on a Sunday, we've had
Even on a
representatives from Moscow, from Norway, and Japan. We very much appreciate you waking up in the middle of the night or staying with us into the middle of the night to follow along.
And with the technology that we put out there, we've actually had people communicate with us while they were on a plane, using the chat and
No kidding.
watching the streams and on the
So what do you think about another
trains as well.
5 meter?
Or at home,
well,
sitting in your
actually, we should
living
have enough
room.
tether, looking down at 80. We could just, you
It's
could
great
peek
that
over
we're
the edge
able
of the
to
ridge, and then we'll
all
just
be
kinda
able to
saddle
watch
up the
this
slope or
wherever
along
we
the ridge from
can.
Yeah, do that
Copy
too.
that.
And we have our OER folks on shore who are also overseeing the Facebook page and the Instagram and other social media. So they have communicated with us with several questions that the public has have contributed. So it's a very
This
broad
ridge
community of
looks like
input.
it's maybe along
Very different from most types of expeditions, and particularly useful
210 or so.
because of the exploratory nature of these cruises. That I think it's wonderful
so you
that
can
the
even just keep
public
Sirius pointed
realizes
at
that
145,
scientists get just as excited as they do,
and we could
seeing
move it
something they
210,
already are
and you just follow
familiar
V2
with or
up
have
the
seen
ridge.
Copy
samples of, but maybe not in the natural
Is that work for
habitat.
you, pilot?
But
Yeah.
also to see things they've never seen and to have this international discussion on the chat room as to what it is we are seeing. And how we could identify it and comparing ideas. It's been very rewarding to see this in the chat room, and I'm in awe of all the knowledge in front of me on the chat room screen.
Yes, me too.
You've made our job a lot easier. Certainly mine. You've helped me because I certainly didn't know these organisms, and I've been trying to learn as we go, and at least I know when I often know something that I've seen before, but I may not know what it is.
Some white crab activity on the far right just went off screen.
See if I can find it back.
Here's our wreckfish again.
Yep, and I did recognize him from previous
Maybe two
Johnson
of them
Sea
arguing
Link
there.
dives,
Oh, yeah.
submersible dives on the Charleston Bump in the early 2000s with George Sudbury's group in the South Atlantic Fisheries Management Council, along with Department of Natural Resources in Charleston in South Carolina. Those were OER cruises in the early days when the office was just called Ocean Exploration. And now they've added research, because so much research comes out of these observations made at sea using this amazing video, but also now with the samples that we collect that are available to anyone who requests.
And it's a way to really move the science forward as fast as possible, because when you think about individual research teams coming out and collecting samples and not being able to identify things, you know, you get this pile of samples. There could be something totally rare and unknown in your pile, and you might not know it for years. But with this kind of telepresence, you can get to exactly what's most important really quickly and know the state of the knowledge just in a few minutes.
And Adrian Copeland just sent us in the chat room a request from one of the audience. Adrian is the OER person I was actually referring to, who is overseeing the questions that come in from social media. And Joe Vaughn asked, I'm sure you have addressed this already, but would you explain why there is so much dead coral on the bottom and what species? And I'll hand that off to my coral expert who sits to my left, Cheryl Morrison.
The species, that's the easy thing to start with, is Lophelia pertusa, and you see some white colonies of live Lophelia coming into the screen now on the bottom. Where you go to different mounds and there's different amounts of live and dead coral, most of it that's really alive is on the top of the mounds. And basically, this coral is building these structures, these mounds that can be, 50, 100 meters off the seafloor over thousands of years. And we're not sure exactly why there's so much dead coral, but it's still really important to the habitat. And this is kind of the usual state. So somebody asked previously, does it have anything to do with like a hurricane coming through or anything like that?
Look at the white puffs.
And we really don't think that has those kinds of storms have much impact on these deep reefs.
Soft corals.
We've talked about the Gulf Stream changing position over time. And so maybe when the Gulf Stream was right over this area, more coral was alive. We're not really sure, but the important thing is that the dead coral remaining in place is what creates this good habitat for the coral that is alive. And so it's all significant in the grand scheme of these ecosystems of the deep here. And if we were to just move off of this feature, we would probably have much less diversity and much less abundance of fauna. When you look at the kind of more sedimented substrates that we've seen on this expedition, you do still see fauna, but nowhere near the biodiversity and concentration of fauna that we see here on these types of larger features.